Chapter 5, Carbohydrate Catabolism. In this topic, the learning objectives are, in general terms, describe the three stages of aerobic glucose catabolism, which are glycolysis, the Krebs cycle, and an electron transport chain, including their substrates, products, and net energy production. Discuss the roles of acetyl-CoA, the Krebs cycle, and electron transport in carbohydrate catabolism. Contrast electron transport in aerobic and anaerobic respiration, and be able to describe the function of ATP synthase and how it interacts within an electron transport chain. So in this topic, subtopics are, we're going to have a general overview of catabolism, we're going to talk about glycolysis, then cellular respiration. So first, an overview. Here we have glycolysis, which you take glucose and you end up with pyruvic acid. Now from here we can go into fermentation, which is in a different topic, or we can go on to respiration. So from here we go into acetyl-CoA formation, the Krebs cycle, and then the electron transport chain. Now this is a nice diagram because it puts it all together for you. Now let's go talk about the parts. Starting with glycolysis, from the name glyco, that's glucose, lysis, so this is glucose splitting. So let's start out with the first set of steps, which all require energy. This is the energy investment phase of glycolysis. So we take glucose and we take an ATP and we put a phosphate on it, making glucose 6-phosphate. So this would be, when we put a phosphate group from ATP onto another molecule, that would be substrate level phosphorylation. Now that we've done that, we got to put one on the other side. So we take another ATP and we phosphorylate the other side of it, forming fructose 1,6-biphosphate. So you're saying, I'm supposed to be getting energy from this. This is a catabolic reaction. Where's the energy coming from? Well, to use a non-cellular analogy, you got to spend money to make money. You got to spend some ATP to get some more ATP out. And this is what's happening. We're investing energy into what was originally a glucose to be able to get energy out of it. So from here, we get the actual lysis of our glycolysis, and we split this. Now I'm going to have to bring up the other side. We get a molecule of glyceraldehyde 3-phosphate, affectionately known as G3P, and we get a molecule of dihydroxyacetone phosphate, or DHAP. To continue with glycolysis, we need to change this DHAP into a G3P, and that requires moving a hydroxyl group, okay, and switching it with a double bonded oxygen. What kind of enzyme would do that? You got it, an isomerase. So we end up with 2 glyceraldehyde 3-phosphates, 2 G3Ps. So you double everything from here on out. You notice we've got 2's in front of everything here, because each one goes through the energy release portion, or the energy generating portion of glycolysis. So we have our 2 G3Ps, and we're going to release a hydrogen, NAD plus to NADH. And because we have 2 G3Ps, we are able to reduce 2 NADHs. Now in the process of this, we're able to take some inorganic phosphate and put one on each end of our G3Ps, and we make 2 1,3-biphosphoglyceric acids. Now here's where we get the energy back. We take the phosphates on the end and we move them from 1,3-biphosphoglyceric acid to 2 ADPs, and we get 2 ATPs. We've just made back everything we invested. So we're breaking even on energy here. What kind of reaction is this when we take a phosphate group from a molecule and move it to ADP? It's substrate level phosphorylation. So here we have just generated 2 ATP per glucose by substrate level phosphorylation. Well, we've still got 2 phosphate groups. So we go through several steps that I won't burden you with to end up with phosphoenolic pyruvic acid, or PEP, and we take these phosphates and we move them to 2 ADPs, 1 per ADP. So we take these and we generate 2 ATPs. I've just generated, through glycolysis, 2 ATPs. So I've made an investment as far as energy goes. And this is what kind of phosphorylation? That's right, substrate level. So we end up with 2 pyruvic acids. What we have here, going back to the learning objectives, our starting product, our starting reactant is glucose. Our ending products are 2 pyruvic acids, and net 2 ATPs. We generated 4 ATPs, but net we get 2 ATPs. We also generated 2 NADHs, which will be important later. We also generated water, but we generally don't keep track of water. Pyruvic acid still has an awful lot of energy left in it. So we're going to go through another catabolic set of reactions, and we're going to synthesize some acetyl-CoA. So the first thing we're going to do is we're going to get rid of this acid group. Now we're going to take this CO2 and we're going to remove it through a decarboxylation reaction. So this is part of why you as humans exhale, because this CO2 doesn't have any energy that the cell can use, so it diffuses out of the cell and it's a waste product. Here is the acetate that results when we take off the CO2, the acid group, from pyruvic acid. So now we want to save this hydrogen. It has a lot of energy in it. So we're going to take an NAD plus and we're going to reduce the NAD plus by moving the hydrogen to it, making NADH. So that means the acetate is oxidized. Then we're going to take a coenzyme and we're going to slap it onto the end in the place of that hydrogen, making acetyl coenzyme A, or acetyl-CoA. To keep track of the energy that we've made 2 NADHs per glucose, because remember we get 2 pyruvic acids for every glucose. So we've made 2 NADHs. The substrates were 2 pyruvic acids, and the products are 2 acetyl-CoAs. So these acetyl-CoAs still have energy in them, so we're going to send them into the Krebs cycle. So the acetyl-CoA is going to come in and the acetyl group is going to be attached to this oxaloacetic acid, making a citric acid. Now the coenzyme A goes off to get another acetyl group. We went from a 4-carbon acid to a 6-carbon acid. So in the process of the Krebs cycle, we're going to knock off this acid group. Once again, whenever you knock off an acid group, we get a CO2. And we want to save that hydrogen. That hydrogen has plenty of energy. So we're going to oxidize the isocitric acid and reduce the NAD plus and get some NADH. Now we still have plenty of carbon here, so we're going to knock off another one and we're going to get some CO2 again. Once again, we want to save that hydrogen, so we're going to take the NAD plus, put the hydrogen on it. We're going to have CoA come on again, and through this process we're going to make an ATP. Then we're going to go on, we've got some hydrogens here that we want to save, so we're going to bring in a new molecule that's going to be reduced by picking up hydrogens. This one is FAD, and it's going to pick up 2 hydrogens to become FADH2. Then we're going to do some more messing around. We're going to oxidize malic acid, create another NADH. Then we're back to oxaloacetic acid. So in the process, the substrates are acetyl-CoA and oxaloacetic acid. The products are CO2, NADH, ATP, and FADH2.